Mindsetters, my name is Megan and welcome grade 12s to today's Life Sciences show. Today we have a special revision lesson in DNA, RNA and meiosis prepared especially for you. We'll be selecting highlights from the lessons shown early in the term to help you revise for your end of term tests. You can download the notes for today's show on learnextra.co.za/live.
Let's go into the actual work then. So the first question we asked ourselves is, where do we find this DNA? In other words, what's the location of DNA? Let's keep with the vocabulary, guys, and as we're going along with the lessons, every new term you get or you come across that I'm mentioning or it's on the screen, take the word down at the back of your notebook somewhere and build up this vocabulary, because terminology in life science is key to whatever we're going to do. So we're saying, where do we find the DNA? And we say that we said earlier, there are two kinds of nucleic acids. We get deoxyribonucleic acid, DNA, and we get a ribonucleic acid, that is RNA. These are the two nucleic acids that we have. DNA is the one we're looking at the moment, is found in the nucleus of eukaryotic cells, where it forms part of the chromatin. Some also found inside the mitochondria and the chloroplast. Now, notice that we're talking about, the other words are more or less simple, but we're talking about this guy here, eukaryotic cells. What is a eukaryotic cell? A eukaryotic cell is a cell that has what we call a true nucleus. In other words, a nucleus that is surrounded by a nuclear membrane, and inside we find the nuclear material. That means chromatin network, the nucleolus, etc. Whereas a prokaryotic cell is a cell, what we would call before, first. And these are what evolutionists and scientists would call the primitive cells. These so-called primitive cells lack a nuclear membrane. They lack a true nucleus. They have nuclear material which is not within an organelle. They are found in a cell. In other words, there is DNA or RNA or both, but they are not bound by a membrane and they're not in an organelle. So the eukaryotic cells are the cells that after, not okay? Where it forms part of the chromatin, right, the chromosomes, and some is also found in the mitochondria and some in the chloroplast. Mitochondrial DNA, chloroplast DNA. We are very interested in this DNA that's found in the nucleus, and later in evolution, we are also interested in the mitochondrial DNA. But as we progress with the work on DNA, as you're going to go along, you'll notice we'll talk more about mitochondrial DNA. So we know now where it's found. Now we want to know how is this thing structured. In other words, what is the structure of this organelle? The first statement there, again a big word, guys. Remember, write the big words down. Polynucleotide. Nucleotide we've already discussed. Nucleotide is a building block of the nucleic acid, DNA and RNA. Both are made up of nucleotides, many nucleotides. If we look at this nucleotide, we can consider the nucleotide to be like a brick on the wall. The wall is made up of several bricks. The same way, DNA is made up of several repeating nucleotides, maybe with similar nitrogenous base or with different nucleotides. Each one has its own sequence. So polynucleotide, many. Poly means many. So nucleotide, poly, many nucleotides. So it means it's a large molecule. A polynucleotide is a very large molecule made up of a string of repeating units called nucleotides. We've discussed that already. Each DNA nucleotide consists of 3 parts. And I told you we're coming back to this now. 1, deoxyribose sugar. Remember, in DNA, we are talking about deoxyribose sugar. We must understand this because when we talk of RNA, we're going to talk about a ribose sugar. So keep that in mind. One phosphate group, and it's not phosphorus, but it's a phosphate group. Keep that also in mind. And one nitrogen-containing base. There are 4 possible bases. What are they? Adenine, A, thymine, T, guanine, G, and cytosine, C. Those are the 4 types of nitrogenous bases one would expect to find on a DNA molecule. Okay? Now let's look at the structure. There we have it. Phosphate, there, attached to a deoxyribose sugar, there, and that is attached to a nitrogenous base. This is one nucleotide. Represents one nucleotide. Now let's look at that in a molecule. There we go. In other words, we're looking at something like this. There, in color, there's it. We're taking this guy out, and we are putting it there. But look at the way it is structured. There we have, again, the phosphate sugar thymine, the phosphate sugar cytosine, the phosphate sugar adenine, the phosphate sugar guanine. They've given you a key for that as well, P-phosphate, and sugar. What sugar we're talking about is deoxy. Guys, remember in your tests, in your exams, whenever you're labeling the sugar in DNA, you must use the words deoxyribose sugar, not just sugar, because there are different sugars in DNA and RNA. That stands to reason. Now, again, going back to the location of this guy that we are talking about. Here's a plant cell, an example of a plant cell. And when we look at it, we'll find that here's the nucleus in this area here. And inside the nucleus, you have the nucleolus, and you have the chromatin network. The DNA is found there. The DNA of the nucleus is found inside the chromosomes. In other words, what happens here is the chromosome is made up of 2 strands called chromatids. And each chromatid, in turn, is made up of large strands of DNA. And each DNA is made up of smaller units called genes. And it is these genes that we started this topic with. These are the genes that determine what you look like. Not look at, Katlego.
Not
look,
what you look at.
like. In other words, whether you're going to have blue eyes or brown eyes, whether you're going to have straight hair or curly hair.
So it's not whether you have black genes or blue genes, right?
No.
Okay,
That's not.
okay.
It's just different genes code for different things. But we're going to talk about that lots and lots and lots about the blue eyes and the brown eyes under the section called genetics later in the year. So this is where we are at in terms of the location. Guys, why do we put a diagram on? Because a picture says much more than 1,000 words. If I show you the picture and I say DNA's location is here in the nucleus, in the chromosome, already you're seeing it. You're seeing that it must register. And obviously, the mitochondrial DNA will be found there, mDNA. And the chloroplast DNA, cDNA, would be found there. Like I told you, we are interested in this guy, and we are interested in the DNA that's found in the nucleus. That's what we are interested in our studies. A little more on the bases. Adenine and guanine are double-ringed structures, and they are called purine bases. Adenine and guanine are double-ringed structures, and they are called purine bases. Thymine and cytosine, I should actually do that and change the color. Thymine and cytosine are pyrimidine bases. and they have single-ring molecules. They are single-ring molecules. The chemistry, you do not have to go into the details. Just you've got to know that cytosine and thymine are pyrimidines, and adenine and guanine are purines. We've learned so much. Let's see if we remember some of it. In your section A, the first question is always multiple choice. 1.2 generally would be provide the correct biological term. And the examiners and markers are sticky. They want the proper biological term, not just a common kitchen term. They want the proper biological term. So we're starting today with some of those. Correct biological term for the following: the base that pairs with thymine. Yep, you're right, that is adenine. And how we know that? At our story. The building blocks of proteins. Okay, we haven't come to this stage yet, but let's just write it down for you. Amino acids. We'll talk about this when we talk about RNA. The bonds that hold the two polynucleate strands of a DNA molecule together. We've just been answering a question on that, and that is the hydrogen bonds. And I would be a little more finicky and say you must write the weak hydrogen bond. You don't even have to write bond because the question says bond. Weak hydrogen is good enough. Repeating units, monomers, that form a nucleic acid. And that is, I spoke about the bricks on the wall. Yes, that is nucleotides. Phosphate, sugar, nitrogenous base. The sugar that forms part of DNA, that's your clue, DNA would be deoxyribose. You don't have to write the word sugar because sugar is already in the question. The shape of a DNA molecule, you will say spiral, or you can say helix, or you can say double helix. They only asked for the shape. So we're talking of the spiral nature, the helix nature of the DNA. So can you see that's your one-word answers? We move on now to the structure of DNA. The diagram below represents a part of a molecule. Study the diagram and answer the questions that follow. There's a diagram there. They're giving you A is adenine, G is guanine. By now you should know that. No, even if they don't give you the key, A is adenine in a DNA, G is guanine, C for cytosine, and T for thymine. And they've given you the molecule there, and they're saying identify the molecule above. Because we did DNA today, it's easy, but in exams you've done DNA, you've done RNA, so it's difficult. But the fact, what you're going to look at, you're going to look at it has two strands, double strand, so therefore this is DNA. There's nothing else in the diagram that tells you anything else, so you can't use anything else there. Label the parts numbered 1 and 5 respectively. 1 and 5. Now, here, there's nothing here, there's nothing here. How do I know this? Wow, what do I do? I'll come back to that. Let's do 1 first. 1 is easy, it's the bonds. So this is the weak hydrogen bonds. 5, now look at 5, look at the shape of 5, and look at the shape of this molecule. Look on this diagram, find another molecule that looks like this, just like a jigsaw puzzle, and there you have that thing there. And therefore, if that was G, then this must be cytosine. I'm just writing cytosine, cyto, you'll know the rest. What is the collective name for part number 2, 3, and 4? Obviously, without even looking at 2, 3, and 4, I can tell you when I use the word collective in a DNA molecule, it would be a nucleotide. Okay. What is the significance of this molecule being able to replicate itself so that the daughter cells have the same chromosome number and the same DNA as the parent?
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision.
So RNA, ribonucleic acid. Remember, we studied DNA, deoxyribonucleic acid. All right. We talk about the differences as we go along. All right. RNA, first of all, is single-stranded. The word polynucleotide we discussed last week, remember? Many nucleotides, so it's a long chain. However, RNA is not as long as DNA. So keep that in mind, because I'm going to ask you a question a bit later. So first of all, it is single-stranded. Secondly, it is long, but not as long as DNA. Second, further on, the nucleotides of RNA differ slightly from those of DNA. An RNA nucleotide consists of a ribose sugar. A ribose sugar. In DNA, we had deoxyribose sugar. A phosphate. In DNA, we had not D-phosphate, we had a phosphate. A phosphate we have in DNA, we have a phosphate in RNA as well. Always look at similarities and differences, because they can ask you for provide the similarities and differences, or provide the similarities only or the differences only. Okay, and they have the 4 bases. In DNA we had 4 bases, here too we have 4 bases. We have either uracil, cytosine, guanine, or adenine. Now which of these guys is different from last week? You're thinking about it? Good. You must have found out now that it is uracil. In DNA, there was no uracil. And uracil in DNA was replaced by thymine. I'm writing it here clearly that it's in DNA, so you do not get confused. That's in DNA. Remember, last week we spoke about the base pairing at cute girl. At adenine always combines with thymine, and cytosine always combines with guanine. So here we do not have thymine, so we have uracil. Keep that in your mind as well. If we look at the structure, there we have it. Again, a picture speaks much more than the words. Okay? There we have the phosphate. Look at the skeleton, the backbone, compared to last week's one. It's the same. Phosphate attached to the sugar attached to the nitrogenous base, and phosphate, sugar, nitrogenous base, and so on we go down. Difference being that there's no other strand on this side, so it's a single strand, number 1. The other difference being uracil there instead of thymine. And as you see, that's the structure of RNA. Now, the other difference is that RNA has several types. But again, somebody may argue, because of the discoveries we've made recently, there are different types of DNA as well. Remember from last week, we had nuclear DNA, mitochondrial DNA, and chloroplast DNA. So there are different types of DNA as well. However, there are specific different types of RNA, and they are, firstly, messenger RNA. Secondly, ribosomal RNA. Notice the abbreviations used. These are standard abbreviations. You can use it in your writing of your answers. mRNA can be used for messenger RNA. rRNA can be used for ribosomal RNA. And tRNA can be used for transfer RNA. Now let's look at each one. And guys, if you listen carefully here, and if you understand where do we find messenger RNA, ribosomal RNA, and transfer RNA, and what each one of them does, that explains protein synthesis. It's as simple as that. If you can understand where we find them and what they do, what's their function, what they do, what are they there for, that, the 3 combined, gives you the whole of protein synthesis. You don't have to worry about a thing if you learn this properly. So let's listen very carefully now. Messenger RNA, first of all, carries information about the amino acid sequence from the DNA. In other words, messenger RNA copies the code from the DNA. That's the first thing. And we're not going to go into detail now because we'll go into that detail later. Ribosomal RNA, they don't have a real shape as such, but they form in the ribosomes, and the ribosomes are formed in the cytoplasm of the cell that make proteins. In other words, ribosomes are the site, or is the site of protein synthesis. Last one, transfer RNA, small molecule, clover leaf shape. it picks up amino acids. Listen to this. It picks up amino acids. From where? From the cytoplasm. And what does it do with them? It brings them to the ribosomes, where they are joined together to form a protein. So tRNA is basically like a taxi. Its job, go to the taxi rank, pick up the amino acids, bring them to the marketplace. In this case, the marketplace being the ribosome, because that is where the transfer RNA need to go. The job of the transfer RNA is simply to take the guys from one point to another. It's like a taxi. The taxi takes you to the shopping center or to the school. But the taxi driver doesn't have to make sure that you shop at the shopping center or that you indeed go to school even. His job is to drop you outside the gate. What you do after that is your business. So the same way, transfer RNA is a taxi. It goes in the cytoplasm, picks up free amino acids, and specific transfer RNAs pick up specific amino acids. Just like when you go to the taxi rank. There are different taxis for different areas. If you go on the wrong taxi, you're going to go to the wrong area. You see? So you've got to be careful how you do that. So the same way, there is a specific code provided for the amino acid, and that amino acid then will go with the taxi or the transfer RNA to the ribosome, and it will combine, and we'll talk about that also just now. Let's look at the diagrams. That's what messenger RNA, just a stranded structure. rRNA, no real, it's mainly coiled in many areas, and this is what they're talking about, the 3 clover-shaped type of RNA, transfer RNA. By the way, the transfer RNA, on this side, it picks up the amino acid, and on this side, generally, you have the nitrogenous bases. And it is these nitrogenous bases that will distinguish between different types of transfer RNA and the amino acids that they carry. There. By the way, when we talk about that, we'll talk about that just now in protein synthesis, so I don't want to say too much there. Are we clear with this, guys? Remember, messenger RNA copies the code. Where? In the nucleus. From who? From DNA. It then takes this message to the ribosome, and it attaches itself there. Ribosomal RNA is involved with the production of the ribosome, etc., and attaching the messenger RNA to the ribosome. Transfer RNA simply is there to pick up the amino acids and bring them to the ribosomes. Remember, we said that the ribosomes are the site where proteins are made. In other words, this will be the factory. This is where the proteins are made, manufactured. This is a manufacturing plant, whatever you want to call it. So that's what we are talking about there. Now, when we're looking at this section on protein synthesis, we break it up into 2 areas. The first area being transcription. Where does it happen? In the nucleus. The second area, or the second process, I shouldn't use the word area here, the first process, transcription, taking place in the nucleus. The second process, translation, taking place in the ribosomes. Let's look at each of this in summary form quickly. What happens during transcription? As DNA replication takes place, messenger RNA forms. How? While the DNA is replicating, when the strands separate, instead of another DNA forming there, messenger RNA sneaks in before the other strand forms, and it copies the code and it moves away. So it doesn't interfere with replication. Replication will still continue. Okay? This is controlled by enzymes. This messenger RNA then leaves the nucleus and it attaches to the ribosomes in such a way that the base triplets are exposed. They are showing. And the base triplets are known as the codon. They are providing the code. What has happened? Messenger RNA has stolen the code from DNA. And now it reveals that code in the cytoplasm. Why is this necessary? It's necessary because DNA, that nuclear DNA, does not leave the nucleus. So it cannot get the code to the cytoplasm. So the only way this happens is when messenger RNA steals the copy or the code and goes out. The next phase is translation. Now the word, again, English is a simple language in certain cases. Transcription means, the verb it comes from is transcribe. And to transcribe means to take down, or rather to copy. So again, from the word transcription, you should know that this is the copying process. Translation, here's a code, we have this code. But if I give you a code and you don't know the combinations, indeed, or you don't know how to, the password for that matter, you can't break the code. So translation means we need something to break this code. If I start talking to you in another language here all of a sudden, you wouldn't understand a word I say. Then I would have to have a translator next to me to do that. So this is what translation means. We're taking this code and we are changing this code to give us something else that we can work with. And that's what's happening here. How does that happen? First of all, the transfer RNA picks up amino acids. Where do they take them? To the ribosomes. And remember I told you earlier, those base triplets, that is known as the anticodon. Now we got two words, the codon, and now we got the anticodon. The codon provides the code, the anticodon breaks the code. But the codon and the anticodon must match. So in other words, if we have here the codon being A, G, C, then the anticodon that comes here should be U, C, G. Remember from our base pairing from last week. But here, remember, look at the special case here. We said at, cute girl. In RNA, this doesn't apply because there's no at, there's no T. So whenever there's A, it will combine with a U. It will not be replaced by a U, but it will be combined with a U. Just keep that in mind. The codon, the amino acids then come next to each other. They join. 2 will be dipeptide, 3 will be tripeptide, many will be polypeptide. And when this polypeptide gets to 50 and more, we have a protein. All of these reactions are controlled by enzymes. And when 2 amino acids join or combine, water is released. This is known as dehydration synthesis. And again, a picture says more. There's your story. DNA molecule is unwinding. Same story as replication, so that's what's happening. As it unwinds, the messenger RNA, the messenger RNA starts forming here with that. Here's the one with the blue backbone. See here, with the C came G, with the T came A. The T was in DNA. We do not say now when there's a T we must put U. No, T always combines with A, and this continues. A, now here, A normally combines with T, but in RNA there's no T, so that is replaced with a U, and so it goes. That's the messenger RNA forming. What does this messenger RNA do? It then moves out of the nucleus. There we go, it's on the left-hand side there, sorry. They move out there and they go out here in this area here, and we have the messenger RNA formed. When it goes out of the nucleus and it aligns on the ribosomes. We go straight to the questions then. Let's look at the first one. I'm only going to look at the correct biological terms. Nitrogenous base, which is found in RNA but not DNA. That one is easy, it's uracil. A triplet of 3 bases found on an mRNA, what we call the collective, we call it the codon because it provides the code. The single strand of DNA that contains the instruction for making a protein, the one that shows the code, we call it the template. The template or the mold. The type of RNA that picks up amino acids in a cytoplasm and brings them, that's easy. Picks up, the word picks up tells you something about what I told you earlier, the transfer RNA. And factors that cause mutations in DNA and chromosomes are called mutagens. The synthesis of mRNA from DNA, formation of mRNA from DNA, this is known as transcription. Transcription.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page. facebook.com/learnextra or on Twitter @learnextra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision. Over to you.
Good. We move straight into the lesson itself, and we start off with some terminology today. First of all, what is cell division? Cell division is a process where cells divide or reproduce. And we look at two types, that is mitosis and meiosis. The cells are the only things that actually are Atik and Indi divide to multiply.
Remember, test him, not me.
No, that wasn't a question, it was a statement. That's why I adjusted both of you.
Oh, thank
Okay. Chromosomes are cell parts, and we learnt about chromosomes when we started with DNA and we moved on, that determine what traits a living thing will have. In other words, what we said earlier, chromosomes are made up of two chromatids. Chromatids are a strand, one strand on the set of chromosomes. And we also went further to say that a chromatid is made up of DNA. So chromosome made up of two chromatids. The chromatids are made up of long strands of DNA. We spoke about this before. Obviously, after replication, a chromosome is made up of two identical chromatids. What are daughter cells? Daughter cells are the cells that are produced at the end of cell division. In other words, the result of cell division. Cytokinesis, cyto coming from cytoplasm, and kinesis, division. So cytokinesis is a division of the cytoplasm, when the cytoplasm is split in two. In an animal cell, this is done by invagination of the cell membrane. In a plant cell, a cell plate will divide the two. We're moving on a little bit more in terms of terminology. Karyotype. Karyo coming from nucleus, type, the type of nucleus. But actually what we are talking about is how are the chromosomes arranged in this cell of this organism. So a karyotype diagram is a diagram of arrangement of chromosomes. Some books call it a karyogram instead of a karyotype diagram. Okay? First of all, we must understand that chromosomes are paired according to size and obviously according to the characteristics that they code for. We cannot have a chromosome that is coding for eye color right next to a chromosome that is coding for bum size. To put it crudely, Indy, I see you cringing there.
The bum size. I was just wondering.
More
if you're gonna start
exercise,
talking
more
about my
exercise.
bum. And you went there. And you went there.
Okay, so the arrangement is called a karyogram, and the set of chromosomes is called the karyotype. The human karyogram, we like to look at that one because it's about us. So the human karyogram shows the 23 pairs of chromosomes. Because we have 46, these 46 chromosomes we have are not just 46 any chromosomes, they are 23 pairs. One set of maternal origin we got from our mother, and the other we get from our father, maternal, paternal. The diagram here clearly shows you a male, normal male, and a normal female. Notice that the chromosomes that are lined up there, they usually start in a karyogram from the largest to the smallest, going down that way. And there you have them numbered, 1, 2, 3, 4, 5, 10, 11, 12, up to number 23, which is not numbered. It's written there X, Y, and in this one it's written XX. Now sometimes in the exams or in study guides, etc., they do not have this there, the XX and the XY. How would you know the difference? And size does matter here. In the one on the right, female, the two strands are identical in size. And in the male, the one is larger or longer and the other one is shorter. So you would know the one that has different bands or strands next to each other, that's the male. And the one that have identical strands next to each other, that would be the female. The other thing we can also look at is that in every set of chromosomes here, there must be only 2. So they must be paired. There you have all their paired except for this one here. So if there's something different, this one here, number 21, has 3 instead of 2. There must be something not right there. So we need to look at that. And when we do that section a little later, we talk more about it. A little bit more about the haploid-diploid story. Remember we said that our normal body cells or somatic cells have 46 chromosomes. Now here you're having a fertilized egg cell that has that. Now I would rather take that out there because you're going to get confused with somatic and zygote. So let's just leave that zygote there with 46. This is the fusion of the sperm and the egg. Where did the sperm and the egg come from? They came from germinal epithelial cells, which are 2N or diploid. One from the mother, one from the father. And when these two come together, you're getting 46 again. So the zygote that develops from there, a fertilized egg cell zygote, will have the full complement of chromosomes. Okay? If this didn't happen, then we would have had 46 plus 46, and you would get something with 92 chromosomes. Now the thing about chromosome number is that it is specific to specific species. Mind the pun on all those specific specificities. All right? In that case, what happens is, obviously there's something wrong if something has that there. And by the way, just as a matter of interest, the number of chromosomes does not mean some organism is more complex or less complex. For example, we have 46 in the 46 chromosomes in our ordinary body cells.
Everyone has 46.
Everyone has 46 unless there's a problem, like Down syndrome. It's a different story.
Okay.
The fern, on the other hand, what Llewellyn was talking about, the fern, moss is going to lead up to the fern eventually. The fern has 300 chromosomes. Now, we thought we were advanced to the fern. Now, in terms of chromosome number, there's no...
What does that mean with the difference? That the fern, I've heard things about the fern.
There's no difference in terms of anything. That is why we're mentioning it here, that the chromosome number is just specific to a particular species. That species, when it procreates, it must ensure that in that species, we must have the same chromosome number. And that is where our friend meiosis comes in. All right. Now, you did mitosis in grade 10, and we're going to go through very quickly those stages because we're going to come back to them later. The first phase is interphase, and if you remember, in interphase, the main story there was DNA replication took place. Interphase, between two phases, interphases. So when that happens, the chromatids were single, and they duplicated themselves, and they became chromosomes. The next phase that follows is prophase, and during prophase, the chromosomes become visible. There's a lot of other things that happen. The spindle fiber forms, the centrioles move to opposite poles in animal cells, and so on. But the main thing is that the chromosomes become visible. In metaphase, middle, the chromosomes are in the middle, at the middle of the cell. In anaphase of mitosis, the centromeres split, the chromatids move to opposite poles. They start moving, rather. And at telophase, we have two daughter cells that look identical to the mother cell, if you want to call it that. Identical. Mitosis, exact duplication. It's like putting a page on the photocopier. You do not expect any distortion. You expect the same copy to come out on the other one. So mitosis is exact duplication. We now look at where we are now, and that is meiosis itself. So what we're trying to show here on this slide is very simple. We're saying, how does meiosis form haploid cells? So they're looking at a parent cell, which is diploid. There you go, diploid. They're just showing you only 2 chromosomes here. Okay? If that has the 2 chromosomes, this is what's happening. There's the 2 chromosomes here in prophase. They're doing something. We're going to talk more into detail about that later. Right now, we just want to know how does the diploid become haploid. So let's focus on that first. So when that happens, eventually after this crossing over is taking place here, you find there were 2 here, and each of these cells have 1 chromosome. So 2 became 1. In other words, it has become half. Diploid is the full set. Haploid would be half. Hap for half there. Diploid, double, but not really being double, but being the full complement. But we use the word 2n and n for that. So understand what we mean by diploid and haploid. Okay? And understand also that what is happening here is the formation of gametes, the sex cells. In the males, in humans, we're talking of sperms, and in the females, we're talking about eggs. And when this happens, this is what is happening. Each one has half of the original content. And that's what we're having there. So that's the first thing we need to understand. How does diploid change into haploid? At this stage, by the way, I just remembered that John sent me an interesting email from Mr. M.T. Aziz. I want to hats off to Mr. Aziz, was my lecturer in botany and zoology at the Transvaal College of Education. He was watching last week, Indy. He watched you and I, and he was impressed.
Fantastic. That's what I want to
His
hear.
daughter wanted to watch Life Sciences in Matric this year, and she insisted her father comes to watch this exciting program. So we're doing something right.
Yes, that's
I
what I
think
like
it's
to
more
hear.
you than me.
Never, never. I think you give me too much credit. It's definitely you.
No, no, no.
It's definitely the beard. The beard.
I'm too old. They're looking. All eyes are glued on you. Could be Belinda too, Indy.
I think it's Belinda actually.
She's
Belinda
in
is
charge
our
of the whole
director.
shoot, so
Yeah, exactly.
nothing goes on without her. There we go. Now, guys, we want to go into the details of what is happening. There we have our parent cell there, diploid parent cell. The first division, prophase. What has happened here? You can see clearly the centrioles forming, centrioles moving to opposite poles, and the spindle fiber forming, the nuclear membrane disintegrating. The chromatin network, which was just like a ball of wool, now becomes clearer as chromosomes. So they contracted, they become like chromosomes. And watch what is happening in this particular stage. In mitosis, we did not mention this. The chromatin became chromosomes. But the homologous pairs need not, in mitosis, lie next to each other. However, in meiosis, it's important that they lie next to each other. And the slide before this, if you remember, we saw this there. This process takes place. What is this process known as? It's known as crossing over. What is crossing over, you ask? Is when the chromatids of adjacent homologous pairs cross over, or homologs, I should say. Homologs cross over. When they touch, this point is called the chiasma. What happens here, guys, is that parts of the one are exchanged with parts of the other one. And when this happens, there's a change in the chromosome structure. And this brings about genetic variety. So let's go back to where we were. So prophase again, all of the normal stuff that happened in mitosis took place. In addition to these two important things, homologous chromosomes lie next to each other, and crossing over takes place. Crossing over brings about genetic variety. All right? Then we move to metaphase. Meta, just like in mitosis, the chromosomes line up at the equator in the middle of the cell. However, in meiosis, the homologous chromosomes are lying next to each other, whereas in mitosis, they were lying apart. In addition to that, in mitosis, there were one chromosome per spindle fiber. Here you can see there are 2 chromosomes per spindle fiber. What follows next is anaphase 1. And during anaphase 1, No centromere, nothing to do with the centromere. The chromosomes, not chromatids, chromosomes, move towards opposite poles, as you can see there. They're moving towards opposite. So half the chromosomes are moving to one pole, and the other half is moving to the other pole. Can you see that this is what brings about the halving of the chromosomes? So already in anaphase 1, the chromosome number has been reduced to half. Also, in prophase 1, crossing over took place, and this brought about variety. So two important concepts here. One is the variety, and two is halving of the chromosome, both taking place in the first phase already. First stage, I would say. Moving to telophase, in an animal cell, invagination will take place. What does that mean? Invagination, moving in of the cell membrane, pinching in of the cell membrane, whatever you want to call it. But it's an inward movement of the cell membrane. Eventually, you can see what happens. At late telophase, we have two cells from the one cell. But these are two different cells. This is also important, because in mitosis, we spoke about exact duplication. We then move straight into, there's no interphase here, we move straight to prophase 2, where we can see the chromosomes. They do not need to contract, they were already contracted in telophase 1. All the other structures are still there, the spindle fiber forms again, and the chromosomes now move to the equator in metaphase 2. Notice here they are lying singly. I want to ask you a question just now. I'll hold on to that. In anaphase 2, the chromatids separate. Why? Because the centromere splits, and the chromatids move to opposite poles. In meiosis 1, we spoke about chromosomes moving to opposite poles. Here we are talking about chromatids. What does this remind you of? Singly lying at the equator, chromatids moving to opposite poles. Yes, you're right, guys. It reminds you of mitosis. So the second meiotic division is like an ordinary mitotic division. Keep that in mind. And they move to opposite poles at anaphase 2, and finally we have 4 cells that have formed, because each one forms 2. There's 2 on this side, there's 2 on that side. 4 haploid daughter cells. Haploid because the chromosome number has been halved, and they are different. If you can look at them, there's a change. There's a slight blue in that pink. We didn't have it before. Why? Because crossing over took place in the division 1.
Well, guys, we've come to the end of today's show. Thanks for joining me. Remember to visit our website, learnextra.co.za/live, to get the notes and to watch the videos. And if you are stuck and you have any questions, send an email to helpdesk@learnextra.co.za, and we will help you. Also, guys, we are dedicating the month of March to Library Month. Join us as we discuss some great books and some exciting competitions on the way. Goodbye.